Yeah, I was just going to say, well, afternoon all. So today we're going to be doing a little bit of a cross section and discussion and a few presentations about the management of BYDV. And this is going to outline something that we've got an initiative here at ADAS to build a kind of BYDV hub so we can develop and learn what techniques have shown in real farm success and what hasn't, and we can use that to help us inform research and decisions moving forward. So I'm just going to do a quick little bit of housekeeping, but I won't hold Daniel up very long because we're here to hear Daniel, Massimo and Andrew's presentations. So quick bit of housekeeping. This webinar we will be recording and that will be made available to attendees with the proviso that if anything that we discuss or anything you wish to flag, if there's something confidential or something you wish to remove, we can make sure that happens. As long as everybody lets us know. Everyone will be muted and their cameras off during presentations, but when we get to the end of talks, there's going to be little time slots for questions, so raise your hand and we'll be able to unmute you and you can freely ask your question then. But if you have any questions throughout any of the chats or presentations, please drop it into the chat and we'll keep a tab on that. At the end of the webinar, we will be sending around a feedback form along with a BASIS link. We haven't definitively confirmed with BASIS whether we'll be able to provide points for this session, but if we can, we will do. We're just waiting back to hear approval from BASIS themselves. So if you provide us with the number, if and when we get that approval from BASIS, we will then ensure that everybody gets any recognition and points that they deserve. So as I say, what we're looking at is management of BYDV. I'm not going to introduce that in too much detail because Daniel is much more suited in his first presentation to introduce the topic. So, but the idea is to see if we can come together as farmers, advisors, and researchers to build together a hub and collectively learn what is working where, how, and the best ways to proceed forwards, and how we can use some of those in-field, on-farm experience to drive research and optimize BYDV management for future years. So if anyone is willing to be part of the hub, we will send a link out with the feedback as well, but also people can contact me directly and we can enroll you on that hub so we can start other meetings and learn what's being tried where. And as I say, we'll be reviewing and we'll send links at the end of this meeting to those who wish to get involved and join. Part one of the presentations today will help us all understand kind of the power of collective thinking and joining and working as a grower hub. So quickly I'll run through what we'll do today. So I've just done the introduction bit, but soon we'll move on in, and Daniel Lebon from the University of Liverpool will give us a short presentation. Then we'll have Nassimo to also Nassimo from ADAS to also talk on some current research that he's doing, and then we'll close with Andrew from James Hutton Institute to discuss the processes and what it's been like working in the hubs that they've worked in. But throughout there'll be various sections where we will ask a few questions and we will raise a few polls. So I believe the first one comes just before Daniel's talk, of which Lucy, I believe, is sharing this. So this is just the first one, just to get the ball running and thinking about people at the start. So kind of how big a problem do you actually see BYDV to you or farms that you manage within your business or within your advisory network? And there are no wrong answers, so please select whichever you deem most appropriate, and then you can hit submit, and then we'll have a quick little run through and then we'll step on to the first talk. Well, that's the appropriate answer that I was kind of expecting to see with people that have joined a BYDV discussion meeting, that yes, you are seeing it as a problem. This is variable across the country, but it's good for us to get a baseline of people's feelings before we start discussing this in more detail. So now we'll move straight into Daniel's presentation. So if I will stop sharing and Daniel can then take over.
Perfect. Thank you very much, Duncan. So yeah, I'm going to introduce BYDV primarily, and then talk about some work we've been doing at Liverpool over the past few years to look at methods to support management of aphids and BYDV in cereal crops quite broadly. So we'll start with a very, very simple, quick overview of why we bothered and why do we care about pests in agriculture. So obviously we're all interested in cereal crops, and we're here for BYDV. Probably know that cereal crops are quite an important crop grown in the UK. So it provides around 25% of our daily calories. Wheat does at least, and then barley contributes quite significantly to our economy. So it's around 1.8 billion annually to the UK. However, as you're all aware, these crops are attacked by a whole range of insect pests that reduce yields, damage the crops, and if go unmanaged and reach quite high levels, can start to reduce yields and damage the crops and ultimately impact food. Sorry, the alarm, we've got an alarm test at 3 o'clock every Wednesday, which I forgot about. So I'll just quickly mute myself.
And sorry, Daniel, when you come back online, can you make your screen full size? Because right now we're seeing the presenter slide, so we can see your future slides as well. So if you can make a big screen, that'd be brilliant.
Sorry about that. Yeah, I don't know why it's done it that way. Let me try it this way and we'll try again. It's done it again. Try it this way. And wait 1 second. Switch screen. Subtitle.
I think if you go to display settings up there.
Ah, perfect.
Yeah.
There we go. That's what we needed. Thank you very much. Yeah, so sorry about the little interruption there. I forgot it's Wednesday. So yeah, there's this nice graphic that I like to show that is just basically crops of the world. And we can see all the cereal crops are in this big section here. So they really are, they're not just important for the UK, they're important globally across the world. And that's wheat, barley, oats, and all the other crops that are potentially affected or attacked by insects and BYDV, which is why we're all here. So yeah, different pests are active at different periods throughout the growing season. And late last year, with colleagues from ADAS, we came up with this kind of schematic diagram that's UK focused, and it shows the period of risk for primarily winter wheat and spring wheat to each of the main insect pests of importance for cereal crops, and then the period of activity for the insect pest, and then the kind of risk window for each insect pest for both spring wheat and winter wheat. And the reason for this is, economic damage only occurs when pests reach a certain density or reach the thresholds we follow. And all of these things all interact to increase a risk window. I'd argue, and I'm assuming most of you would probably agree, that aphids and BYDV are some of the more damaging or potentially damaging insect pests and diseases we have to deal with in the UK. And the reason for this is really the feeding behaviour of the aphids. So this is just our final overview slide here, and there's around a dozen or so different barley dwarf species present across the globe. In the UK, we primarily focused and interested in BYDV PAV and BYDV MAV. And these are two related species that are primarily vectored by the grain aphids or the cereal aphids we have in the UK. So that's the grain aphids, Rovidavini, the Berteriot aphid, Arpaedi, and the rose grain aphid, Metapalobium dihodum. They're all vectors of BYDV PAV and potentially vectors of BYDV MAV as well. So the way the disease cycle works is that the aphids have this really cool feeding behavior. So they have this needle-like mouth part that they use to penetrate the plant tissue, and you can see it kind of here. It's like this little feeding needle, and they use this needle to penetrate the plant tissue, and then the feeding apparatus goes all the way through the different plant layers until it reaches the phloem, And the phloem is basically the sap in the plant that helps with nutrient transport from the roots up to the shoots and back down. So that's where the aphids feed from, and they're basically, it's a very sugar and nutrient rich sap, and the aphids feed from this and directly siphon away plant sugars and plant nutrition. And that can lead to around 5 to 20% of yield loss. But the real important thing, and that's why we're all here, is Barley-Yellow Dwarf Virus. So because the aphids have this really unique way of establishing a feeding site and interacting with the plant, it makes them really efficient vectors of agricultural diseases. So I like to think of them like the mosquitoes of a plant. So if you think of a mosquito, it's got this needle-like mouth part. It can bite you, it can reach your blood, and if it's carrying a parasite, it can infect you with the parasite. It's no different with the aphids. If they're carrying BYDV, then they stab a plant, reach the phloem and feed. If it's a cereal plant, they'll infect the plant with BYDV. So the way the disease cycle works is, we probably all know that we get primary infection, and this is caused by migrating aphids coming in from the landscape into the field, and the aphids usually pick up BYDV from somewhere in the landscape. They'll infect the plant they're feeding on, and then they'll start producing offspring. We get a cyclical period of these offspring developing into adults that produce offspring that develop into adults. And when the density gets high enough, these adults will produce offspring that will develop back into flying adults. And these flying adults will then fly off and cause extra primary patches. And then the walking or crawling adults, what they'll do is when the density of aphids gets really high or they get disturbed, they'll just fall off the plant and then they'll walk and you get secondary spread. So there's two main ways that the virus is spread through the crop. We have this primary infection and the secondary spread, and then we get these patches of diseased grass, diseased crops within the field. That's quite devastating. So the way that a lot of us will look at pest and disease management is to consider the IPM pyramid. And obviously that can be structured into different layers. And what I'm going to talk about for the next kind of 10 minutes or so, 15 minutes or so, some projects we're doing at Liverpool to look at how we can help BYDV in insect management by helping refine and develop techniques to help with this pyramiding way of managing the pests and the disease. So we're splitting it into two parts. So part one is an overview of some insecticide resistance monitoring we've been doing at Liverpool for the past 2 years. So it started 2022, 2023, and it's coming to the end. So we're providing overview of that, an overview of that, sorry. And that's basically what we found, the 3 main species we've tested, and we'll get some insights there with what that might mean for decision support and how that can be integrated into making management decisions. And then the second part is we have a smaller ongoing project that's looking for screening for Barley-Yellow Dwarf Virus tolerance in some barley germplasm. And that really fits right at the bottom of the pyramid with kind of developing resistant or tolerant varieties. So we'll discuss a little bit about work we're doing there. And then right at the end, I'll give an overview of some other BYDV projects we've got going on. So you can see that we're not just doing one thing, we're trying to look at this from lots of different aspects from across the IPM pyramid and from across the way of managing BYDV. So for monitoring the insecticide sensitivity, as you can probably expect, we focused mainly on the 3 main cereal aphid species we have in the UK. And we've sampled these from across the UK since in 2022, 2023, and last year. And what this has involved is basically working really closely with agronomists, farmers, and ADAS to coordinate sampling efforts, where they'll volunteers, farmers, ADAS staff, and colleagues will go out to the field, sample some crops, sample some infected tissue, and send those aphid lines to us at Liverpool, where we try to establish them in lab cultures, develop a pure culture for each aphid line. So it's all come from one aphid individual, and then test these for insecticide sensitivity or resistance to pyrethroids. And the overall idea here is to give some insight and input into the development of potential decreased sensitivity in the bird-cherry oat aphid and the rose-grain aphid, and to give a snapshot overview of the current status of pyrethroid resistance in Sitobium avene. So there are 2 main ways we've done this. So for bird-cherry oat aphid and the rose-grain aphid, we've used the leaf dip bioassay. And this is a really simple assay where essentially we cut up sections of barley leaves and we prepare a series of insecticide concentrations ranging from field relative concentrations, so a stock concentration, all the way down to, I think it's 10 to the -4 of field rates. That's minimal amounts of insecticide under control. What we do is we dip these leaves in the solutions for around 10 seconds, put these on a plate and then add a number of aphids, move these through a growth chamber, and then monitor the effect the insecticide and the concentrations have on the aphid responses, and then score the aphids as alive or affected. And this broadly follows established IRAC methodology. And then for the grain aphid, we focused primarily on screening for the knockdown resistance mutation. So the way the pyrethroids work is that they target the nervous system of the insect and basically render the insect, the aphid, immobile, and then the aphid can't move, it can't feed, and then the insecticide essentially slowly but surely kills the aphid. But the grain aphid and the peach potato aphid have all developed resistance against this, and this has been linked to specific genetic regions within the knockdown resistance gene, and we can screen for these in the lab. So that's the approach we took for the grain aphid, just because it's a little bit more straightforward, it's a little bit faster, and it let us screen for a lot more pardon me, populations. So our main results here are for the 3 aphid species over the 2 years or 3 years of the project. We sampled hundreds and hundreds of aphid populations, but in total we were able to establish and test 89 serial aphids across the 3 species across the years. And these graphs just show the kind of spatial distribution and spatial spread for the 3 aphid species. So for each species we have samples from England and Scotland. And then obviously for Cetobia navini, it's a bit more abundant, it's easier to find. Have a lot more samples for Cetobia navini. So I think we've got something like 11 for rose grain aphid, 19 for the Burcheriot aphid, and around 60 or so for the grain aphid. So what I'll do over the next few slides is go over this in a little bit of detail for each species, just to give you an overview and an insight into what's going on. So for the grain aphid, we focused on knockdown resistance. And in total, over the duration of the project, we tested 59 populations from across the UK. And I was quite surprised to find that we got quite low levels of knockdown resistance prevalence across the populations across the different years. So we only found 4% of the populations had knockdown resistance in 2023 and 9% in 2024. In 2022, we only sampled from, I think, 1 or 2 fields, and we got 0 knockdown resistance in those samples. Obviously, the caveat here is that we're only testing the areas where we've got aphid samples from, so it's not fully representative of across the UK, but it can give us a good snapshot and a good overview of what we think is out there. And the prevalence is quite low, which I think is good news. And one thing we did to look at this in more detail is for 35 of these populations, we genotyped them. So by that, I mean, we took the aphids and we looked at the genetic diversity. And the idea behind that is we know that there's some grain aphid clones that are insecticide resistant, and we know that there's some that are characterized as insecticide susceptible. And what we wanted to do was to give an overview and say, okay, if any new or any other clones of resistance evolved or developed, or is it still just linked to this one clone we know? And for the others, just generally what's the diversity of Cetobia navini like in the UK? So we sampled and genotyped 35 of these populations from the 59 we tested. And I guess kind of good news is that there's still only one resistant genotype out there that we can detect, and that's the SA3 genotype here. So the SA3 genotype, all aphids that were resistant or had the resistance allele belong to the SA3 genotype, so there's no other knockdown resistance developing among other aphid genotypes, which is good. But that was the second most abundant genotype, but I think that needs to be caveated against the fact that it's quite a diverse species. We get quite a lot of genetic diversity within the populations, which is interesting in itself. And that got us thinking we wanted to look at something in a little bit more detail. So one thing we had access to is I've got a colleague in Liverpool who has a 81 plot spring barley trial over at the Stockbridge Technology Centre in just outside Selby. And what we wanted to do is, over one of our years, we sampled across this trial, and in total we collected 14 populations from this one field trial, and we screened them for knockdown resistance and genotyped them, with the idea being that we wanted to see how diverse are the aphids within a field and what could that potentially mean with regards to management and control. So this is just a quick overview of what we found. We found there, and just to also highlight, we've got a PhD project ongoing. We sampled well over 200 grain aphids from across this field, and he's got plans to look at KDI distribution and loads of other factors that can give us a finer spatial temporal look into diversity within the aphids across the field. So what we found was essentially we got quite a big mix of populations. So there were quite a high number of populations in total. So I think we've got 8 genotypes at least identified. So what this little schematic here shows is it's just a 9x9 grid that just gives you an indication of this would be plot 1, this would be plot 81. And what's highlighted are plots where we collected and successfully reared an aphid, and then the lettering and the colouring just denotes the genetic diversity. So we can see there's a mixture of genetic populations across the field, which I think is quite interesting. And what's even more interesting is this SA3 population. That's the aphid, the grain aphid genotype that has characterized knockdown resistance. And although we see that present within the field, it's not spread across the field. So there's possibly some sort of fitness consequence associated with having that resistance mechanism that's preventing that aphid from just proliferating across the field, which I think is probably good news, because it means that it's not going to completely derail chemical control if it doesn't proliferate widely across the field. And just to flag up that we've got some student projects looking at this in more detail. So we've got a PhD student trying to link this genetic variation with different fitness traits, and we've got a master's student looking at virus transmission capacity and efficiency within these different aphid genotypes and aphid populations as well. So hopefully that'll help give us a little bit more insight. And then briefly moving on to the Burcheriot aphid. So 19 populations tested. So the graph we have here is, I won't go over it in a lot of detail, but other than saying that along the x-axis on the bottom here, we have our insecticide concentration. So that ranges from 0, which is our control, up to 10, which is equivalent to our field rate application. And then we have our number of aphids affected in total on the side axis here. And obviously 1 means all the aphids died. And we see a nice kind of gradual increase in the populations there. And just to highlight these purple square populations here, that's population SA44, which is a insecticide susceptible Cetonia viniculture. So that's our control. And then these little blue circles here is population SA3, and that's the insecticide resistant Cetonia viniculture. So we can see that there's resistance there as well. And we broadly see that the majority of Rupulus hyphen-paidae we tested were fully controlled at the field rate. So 17 of the 19 populations were. Where we need to highlight that there were 2 populations, both sampled from Yorkshire in 2023 and 2024, that didn't have full mortality at the field rate concentration. So one had 90% and one had around 97%. So there's possibly reduced sensitivity emerging there. We need to look at it in a bit more detail. They saw similar things in Ireland a few years ago, and then it disappeared. So it's possibly metabolic, but we're genotyping them to see if it's linked to genetic mutations as well. And then the good news for the rose grain aphid populations are everything was susceptible. So on this graph, we have highlighted the susceptible and resistant controls again. But fortunately, all of the rose grain aphid populations we tested were fully controlled at 10% of the field rate. So there's no decreased sensitivity emerging there. There's some variation in responses, but that's probably just biological variation within the system. So brief summary of the pyrethroid data before I quickly move on to talk about our tolerance. Pyrethroid resistance in grain aphid is quite low, so we found around 4 to 9% in the 2 years we surveyed for this. But the populations are very diverse with multiple genotypes occurring within a field, and this could have other kind of more intricate effects on aphid spread and aphid feeding impacts. We possibly see reduced sensitivity to pyrethroids in the Burcheriot aphid from 2 samples sampled from Yorkshire. And this adds to recent reports in Ireland, China, Germany, and Australia around similar levels of control as well, around 90, 95%. rose grain aphid better outlook, are susceptible fully to pyrethroids. And I didn't mention in detail because we focused on BYDV and grain aphids here, but we've also been screening peach potato aphids, and I thought it would be interesting to highlight that we see very high pyrethroid resistance in the peach potato aphids we tested. So I think we tested around 35 to 40 populations, and 76% contained at least one form of pyrethroid or insecticide resistance mechanism. So the story with peach potato aphid is completely different than grain aphid. Insecticide resistance mechanisms in the peach potato aphid are really highly prevalent in the populations. So just very briefly, the second part of the work we're doing at the moment is screening for BYDV tolerance in barley germplasm, and this comes over under the prevention aspect of the IPM pyramid. So a lot of organisations and a lot of work has gone into looking for aphid resistance. So there's work being done at Rothamsted on aphid resistance in wheat, and there's work ongoing in James Hutton Institute for aphid resistance in barley. And for barley at the James Hutton Institute, they've found evidence that there's a specific compound that's possibly involved. But this is always one part of the story, and I'm always left asking the question, but what about BYDV? So I've been speaking with a collaborator up at James Hutton Institute, Joran Boss, who's got a PhD student screening some of these barley varieties that differ in this defining compound for aphid resistance. And what we're doing at Liverpool to complement their research is to screen these same lines for virus tolerance as well, to see can we find dual resistance or tolerance traits that are active against aphids and BYDV. So I thought it'd be interesting to give a brief insight and overview into that. So the way we're doing this, it's all in controlled environments at the moment, and then to get a look-see to see what's going on, and then the long-term view would be to take it out into the field. So what we're doing is essentially we're growing the plants up to kind of the seedling growth stage and challenging them for 96 hours with BYDV carrying aphids. We then insecticide treat these, let the plant incubate for 6 weeks and screen for virus symptoms, detect the BYDV using ELISA, and measure and collect some physiology data. And that helped us look at both resistance to the virus and tolerance. And this is just a quick overview of what we found. So these graphs, everything is the physiology of a BYDV infected plant relative to a control plant. And the top one here we have plant height, and the bottom one we have plant mass. And along the bottom of each graph we have plant variety. It's just a random number. CS is our control susceptible, and C2 is our control tolerant. What we're interested in is the dashed red line. So the dashed red line is at 1, so that's no difference in the physiological responses we've measured in the BYDV challenge plant relative to the control plant. So we see for plant height, with a tolerant variety, we get an increase in plant height in the virus challenge plant, which possibly indicates some sort of physiological compensation mechanism. And then for the susceptible plant and the plants that are do less well, we see a reduction, so we see a negative effect with BYDV. And this is something that we're just quickly looking at screening, and it's really just a preliminary study at the moment, but we're hoping to use this germplasm to see if we can identify anything that could be useful. And going forward, we're wanting to repeat this to look at a broader range of plant responses to BYDV and try and take it from the glasshouse to the field, and also look at whether we can identify some of the biochemical and metabolic processes that are driving this as well, if we do see differences between the varieties, because I think that would be quite interesting information to know. And then finally, I thought I'd briefly highlight some other BYDV projects we have ongoing at Liverpool. So we've got one that's looking at variation in virus transmission efficiency between different aphid populations, and we've been doing this for bird cherry oat, grain, and the rose grain aphid. So we're testing different genotypes. For grain aphid, we're testing both common and rare genotypes, and for rose grain aphid, just different populations. We're also looking at identifying the processes that allow these different aphid populations to be excellent or less efficient vectors and transmitters of BYDV. That'll hopefully give us a little bit of insight. We've got some modelling work ongoing with some colleagues up in Scotland, and that's putting together loads of data we've generated over the years on aphid development, reproduction, dispersal, and virus transmission, and working with some modelers to develop a simulation model where we can predict the spread of the virus and the dispersal of the aphid across a field for a given aphid genotype. So that would be interesting to look at for insecticide resistant versus insecticide susceptible aphid populations, for example. And then we've got a student project looking at whether soil or whether plants in inoculated soil bacteria, with soil inoculated with beneficial plant bacteria, can provide a sustainable method of managing and tolerating BYDV as well. So that is everything from me, and that was a whistle-stop tour, I guess, of our insights into insecticide monitoring and a brief overview of what we're doing and the work we're getting started with BYDV tolerance.
Excellent. Thank you very much, Daniel. And I must say, very well done. You timed absolutely perfectly.
Oh, we try.
It's good experience. Well done. But yeah, some absolutely fascinating work there. So if anyone has any questions, just raise their hand. We've got a little moment now before NASA moves, because we've got another couple of questions we'd like to ask you, but we'll give you the floor first. So we've got a question here from Neil. So Lucy, can you unmute Neil?
Sorry, Neil. Yeah, you're unmuted now, so you should be able to unmute yourself.
Right, can you hear me
Yeah,
now?
Yes,
Yeah, we got you.
you know.
Okay, just a couple of points. One was, I did see a recent, I think it was Rothamsted presentation suggesting, in the past it's been MAV or PAV, and the variation between year has varied up and down depending on the season. But I think some recent work was suggesting that PAZ is becoming the most dominant one, and that's the most dominant one within Europe. So just as the first point is just as an awareness, because most of your testing on the last slide was MAV and PAV.
Mm-hmm.
Do you want to comment on that?
Yeah, so that's probably work from Lawrence at Rothamsted. So
Yes,
I've been
that's right.
speaking and working with Lawrence at Rothamsted and Louise at Cheggusk. And they've both been really instrumental in developing new molecular diagnostics, because PAV and PAZ are very related. And it's a question that we have as to whether we're seeing a change in PAZ dominance or whether it's always been PAZ, but we've just not had the molecular tools to be able to detect it.
Yeah.
But we are working on PAV and MAV at the moment, and that's just because we're going off what is currently thought to be the most prevalent. And obviously, if PAZ does turn out to be more dominant, then we need to switch and start working with PAZ. So I mentioned some of the comparative virus transmission studies that I'm working on. One of them is in collaboration with Louisa Cheggusk, and we are screening our rose-grain aphid populations for PAZ transmission as well as MAV transmission and PAV transmission. So we're aware of it and we're starting to slowly integrate it. And the reason for focusing on MAV for the grain aphid is that the grain aphid is not very good at transmitting PAV at all. In the experiments I've done, I found that the grain aphid transmits PAV with around less than 10 to 15% efficiency. Whereas if you compare this with MAV, it's 80+% efficient at transmitting it. So I think that's a nuance that we just need to be aware of going forward, is that it's not just the presence of the virus, it's the presence of the virus species and the vector species, and possibly even the vector genotype, and all of these things are interacting to determine a certain level of risk. So I think we're all aware that it's not as simple as we all believe it is, and we're trying to work together on kind of acting on that.
Can I just answer, coming back on that one, the reason, again, PAZ was dominant, or the importance, because it's more, shall we say, damaging as a virus relative to the other two. And can I just, while I'm on, can I just ask one other question in terms of when you're looking at variety susceptibility, right? I'm assuming you're looking at susceptibility in the non-tolerant sector. Are we talking about that? Because I mean, just about every winter barley coming out in France now is obviously 6-row. It's BYDV tolerant. There have got now some 2-rows, and there is some 2-rows in terms of spring barley as well. So is it the tolerance you're looking at in the non-tolerant sector, the variability? Because obviously, again, Avalis did show us a lot of variability within that as well.
Yeah, I'd have to double check with the collaborators at James Hutton. They just sent us the seeds randomly labeled, so I need to check with them what that all matched up to and kind of whether that's a mixture of, I know that it's got a mixture of some commercial and some progenitor species. So we're looking at kind of the wild relative effect as well. But within there are some commercial varieties, and I'd need to double check with them whether that's just, whether that's a diverse grouping across all the different barley varieties and types we have, or whether it's just, a group. I'd need to double check. I'm not 100% sure.
The way the French did it, they looked at the impact of an insecticide applied on a variety and the effect on yield. So the uplift in terms of yield. So it was the same varieties across the same variety trial, and look at the uplift. I mean, this was done over 10 or 15 years ago. Anyway, sorry, I've answered all my questions. Thank you.
It's all right. Thank you.
So, we've got another one now, which we'll try and squeeze in if Sasha can be quick before Nasimu's talk. So if you can allow Sasha to unmute himself, Lucy.
Sasha, you can unmute now.
Thanks all. Daniel, very nice presentation, and some really interesting findings there. Did you see any regional variation in the percentage of Satobi Navini grain aphid that had resistance?
That's what we need to look into next, because we need to look at it with the caveat that there are some regions like Yorkshire where we've got a lot of populations, and then there are some regions where I think we only have a handful from Scotland and from the southwest. So
Okay.
we need to look at kind of, what's the likelihood of, if we've only got one population from that region and it's got KDR and it's going to be 100%, then that's probably not representative of the full diversity of what's going on. So we need to look at it in a bit more detail, a bit more nuance to kind of answer that.
Okay. There's Steve Foster's monitoring that that information is available on the IRAG website through the kind of on the cereal. The resistance issues in cereal pests, that's picked up quite a lot of regional variation. So sort of 71% of aphids with resistance in York and 2% in Star-Crossed. But obviously, again, there's big differences in the actual sample numbers. One other question is this data being published?
It's in the process of being published, yeah. So Nassama and I are working on putting that together. So it will be available and it will be out published in some point in the next few weeks, month or so. I think we're meeting on Friday to discuss it actually. So it will be at some point. Just one thing to note with with Mysis persicui, for example, we saw the majority of resistance on rapeseed crops. For the other crops, we saw less apparent resistance. So we sampled rapeseed potatoes and some others. And when we saw about 80% of the populations that were resistant came from rapeseed, and the rest were just kind of evenly distributed across the crops. So that might be something else to consider alongside the regional variation as well.
Interesting. Okay. Thanks, Daniel.
Right, excellent. Thank you very much, Daniel. That was fascinating. In context of time, I think I'm actually going to skip past a couple of polls that I did quickly have. And we'll try and pick those up later on. But I'd like Nasimu now to be able to put his microphone and camera on and then share and present work that he's currently been involved with. So the floor is yours, Nasimu. You're still muted at the moment, Nasimu.
Okay. Can you see my screen?
Yeah, everything's coming through. Excellent.
All right.
Crack on.
All right. Good afternoon, everyone. I'm Nasimu Musa, and I am an entomologist with ADAS. And today I'll be talking about part of what ADAS has been doing in contributing to the effort in managing BYDV. And one of it is in the area of pest thresholds and also in-field assessment of pests So I will go straight into my presentation. For today, you will expect to have a summary of BYDV. I will not go over what Daniel has said. I'll just skim over that quickly, and the vectors as well. And I'll also be talking about pest treatment thresholds, primarily as BYDV vectors, aphids particular, and also the pest detection models we've been developing in a project, and what we think of their current application in trying to assess for pests in the field. If there's time, I'll also talk about an emerging pest and cereals that we're currently considering, and also put it out there just to see if you've been seeing any related symptoms, and open for discussions as well. So BYDV, it's a pest, it's a virus that is transmitted by aphids, and it's of the Letuviridae family, and it's important for cereal crops like barley, wheat, and others, but also infects grasses, and they cause symptoms on plants, which include stunted growth, yellowing, and reddening of the leaves, which start from the tip. And again, this could reduce tillering and cause very significant yield loss on the crop. So this image that you have here is an image that we collected at ADAS, and you could see the reddening of the wheat plant on the left, and also the yellowing of the barley plant here, which typifies the symptoms of BYDV on cereal crops. And for the vectors of BYDV, the association with BYDV and the aphids is in a persistent manner. So the aphids, once they carry the virus, they retain the virus in their system throughout the period of their lifetime. So, and they are not able to lose it at any point, so which makes it very transmissible. And two key aphids of interest, which are the Bacteroides aphid and the grain aphid, and both are important in all regions that cereal crops are grown. And for the aphid life cycle, just a brief overview. This might be small on your screen, but I'll try to run through that very quickly. I'll start the aphids. Very complex life cycle, and you have from the eggs, they overwinter as eggs or as wingless aphids, and while doing that, they don't have much consent. They are not much of a concern at that stage. But as the weather begins to warm up in spring, reproduction starts, and they pick up in reproducing pathogenically. They bulk up, and when they are so many, that triggers them to develop the wing stages, which that time is warm enough, and crops again are out in the field, and they migrate into crops. And if they carry the BYDV, able to transmit the virus. And as Daniel said, secondary transmission again, once they spread within the crop. And why they're important in the UK is also because during in autumn season, that's when crops are very young and out in the field, and then those aphids that have that wing stage can also get into the crops and again spread the virus, which complicates the integrity of the plants. So we have been considering pest threshold for a while with ADAS, and as Daniel's summary suggests, we have been looking at the range of pests, and of course aphids are one on our radar as well. And just a schematic overview of what pest threshold we consider as pest threshold. So on this graph here, you have pest numbers, and they increase in their numbers up as many as they can, as the plants can take. And again, this x-axis shows the time, also the crop growth stage. So the green line you can see is the increase in the numbers of aphids with time, and the red line shows the particular threshold for a pest. And once there's an intersect between those two, that's a trigger that the pest threshold has been reached, and depending on the crop growth stage, it could warrant treatment or not. And for BYDV threshold, there's really no universal pest threshold available. I think what it's commonly believed is if the presence of aphid and they're increasing in numbers, you should already take measures to go out to treat your crops. Again, for BYDV management tools, there are tools that can help you in monitoring that, and like the Acrobat model by ADAS and the TSOM models by HDB as well. And I believe the TSOM model is also on the IPM Decisions platform, so you could easily get an alert on when to go out to scout your field for aphid. So these tools are important to sort of trigger when to go out to monitor for aphids. And on the back of that, we have been considering ways on how to help in the detection of pests. So once you have a trigger on a pest, for you to go out to the field to go detect, to scout for pests, and aphids sometimes could be very challenging for some individuals. And that was the thought behind developing tools and for help aiding with the detection of such aphids, and not only aphids in cereal crops, other pests of cereals as well, and other crops as well, and their pests in all pests and crops. So that's the main aim, one of the aims rather of the project that we've been developing together with Sheffield University and Liverpool University, where Daniel is, and then Mutus as well, which is a software development company. And for the pest detection workflow, we have image collection, video collections. We've supported with that at ADAS for developing that, and our partners have annotated the data, and our partners have developed the models, which I'll be showing some of what the models have, performance of the model today, and what we also think this will be, how we can apply this for BYDV management. So for pest image collection, so across the whole for the project, we've collected close to 30,000 insects, so including beneficials as well. So we have a range of pests in our database. We have close to 300 videos as well, and we have key pests that we've collected most images as well from, and these include grain aphid and bacterial aphid, which are vectors of BYDV. And just to show you, I think this video was playing when I played this, came on the slide. So these are videos collected as well. So gone out to the field. This was a field near High Mothorpe where I walked from, and it was heaving in aphids, and it was just a good day for us to go collect aphids. We could easily spot them and collect images of them. So we had a number of our collaborators come from Sheffield to come collect images of pests. And we also had public databases of aphids as well, and which we use in our model development. And also we had cultures within our facility of aphid, grain aphid, and bacterial aphid, which were images we collected from them and used in developing the model. So we had two main model conditions, and I will explain why these conditions were key. One is that we require that they are able to have the capacity to detect several pests. So we want a model that not just detect one pest accurately, but be able to detect a range of pests with good accuracy. And secondly, the conditions we had was that they are lightweight. You don't want a heavy model to put on your phone, which will take ages for it to run. So we wanted something that could be lightweight and effective in detecting the pests in the field. And so this cluster of images here just shows you how the model has performed in detecting green aphid and once we tested it, and other pests as well. So the accuracy has been for 76% for green aphid and 65% for barbiturates aphid. So the model, skip that, is YOLO version 8T. So that's a model that has the best performance when we compare that against several models tested during the project. Again, our model again you could see has a light, can perform in a light detection function. So once we have a pest we want to identify, and then we can go focus on our pest and be able to tell what pest it is in the field on cereal crop and also on oilseed rape. So this just shows you that we have wider scope on our performance of the model. And I could talk about how this could be applied next. Before that, again, in the course of the project, we not only just wanted to have function to detect pests, but also get contextual information from the point of image collection. So information about weather, information about humidity, and for the agronomist or the grower, and it could feed in what growth stage of the crop that they're observing. And once it's the count of aphids, for example, barbiturates aphid have made 5 plants per 5 aphids per plant. So you could, the model again could be able to generate suggestions that the agronomist or the grower can be able to use in deciding whether to take an action or not to take an action. So, but the model again gives you information on what was used to generate such advice or suggestions. So the thinking behind why the particular suggestions were made. So you can go through that as well using the as an output from the model. And there are 2 sets of actions that the model could also display, and that's an immediate action. So one of it is, for example, you could want to consider to spray with the level of aphid that you have in your crop or not. And future actions as well. So what could you do? Do you want to have systems where you could encourage the beneficials or aphid predators around your vicinity of your farm? So these are things that growers may want to consider in the future as well. So this information again, we're all sourced from materials from HDB and other internal materials from ADAS as well, to make sure that what is being generated is not out of it's not out of context or out of what could be reasonable. And to conclude on this technology, cereal aphid again remains an important vector of BYDV, and assessment and identification are key for making decisions. And we think that mobile data today with the invent of AI could be used in scouting crop and helping in identification of what pests or what is not pests in the crop. And with the life detection technology that we've shown you earlier, I think it could also be incorporated into farm machinery systems where a decision to spray or not to spray when walking through a field, or when a machinery is passed through a field, that could be useful. So that spray when that could be the idea could be that spray when pests are present and don't spray when beneficials are there. So that's something that could be applied. So this year we'll still be conducting more field testing of this app and functions of the model. So if you'd like to participate, please do contact me at Nassama Musa at ADAS, the CEO of the UK. And that's quick on that. But before I take questions, I just wanted to chip in on the emerging pests that I talked about in my outline. On doing that, I just want to say at ADAS, we have the pest evaluation services that have been running for many years. And we have a team of experts that can help you identify pests. So do get in touch. If there are anything that come up in your crop and you want answers for, we could easily provide that from the range of expertise we have at ADAS. Now for the emerging pests, what we think is a root knot nematode, which we have been seeing for a couple of years now, damaging the roots of cereal crops. And once I tried to look at galls and extracted its females, which has this sac of eggs there, and we could see the eggs, photographs of the eggs that were extracted from the sac here from the root knot nematode female. And this is an adult male from what we've seen from this root of the cereal. So we think there's not much understanding of this pest in the UK. I just wanted to also know if have you seen such pests? Have you seen such symptoms, taunting or plants and roots with such goals? Please, if you do, please just get in touch and we would like to confirm what they are. So again, plug in what ADAS can do as a conclusion, and please, happy to take questions.
Excellent. That was really interesting, Nasamu. Thank you very much. If anyone does have any questions, we'll at the moment we'll hold off until after Andrew's talk, just conscious of time. So we'll bypass my questions to you all until after Andrew's presentation. But if anyone wants to drop any questions within the chat, we can address them as is, or as we go through. But really interesting to see that kind of development of using modern technology to be able to identify pests and beneficial simultaneously. So watch this space. That sounds really exciting. So if I can get you to stop sharing your screen, Nasamu, and we'll move straight into Andrew's presentation. So if Andrew, if you can unmute and share your screen and your camera, please. We'll move straight to your presentation.
Okay, thanks very much, Duncan. Let's see if I can share. So hopefully you can see my screen.
Yeah, you're all good.
Great, I'll get started then. So thanks very much for asking me to talk. I'm Andrew Christie, agronomist and agri-tech specialist at the James Hutton Institute, and now field manager as well. But I'll be talking today about how we demonstrated IPM in action through the IPM Works project, and our approach to working in a farmer hub. So first of all, just to give a bit of background to the project, IPM Works was a European-funded Horizon 2020 project, included UK partners James Hutton Institute, ADAS and LEAF, and an Irish partner, Teagasc. 31 partners overall, covering 16 countries, and adding to existing national farm networks with 22 new hubs or farmer clusters, with the aim of improving pesticide reduction techniques, and doing this through demonstration and knowledge exchange activity for integrated pest management best practice. So looking at the project's overall objectives, the aim was to reduce reliance on plant protection products based on an initial EU target to reduce overall pesticide use by 50%, which is rather ambitious. So each partner in our own context was tasked with taking a regional approach. So taking into account for the east of Scotland with Dundee James Sutton Institute being at the heart of that, we're looking at a arable focus, a wet, cool climate, limited cropping options dominated by malting barley, and a preference for traditional farming systems, which means the plough is king currently. So as a hub coach myself for the James Sutton Institute, I was tasked with bringing together a group of farmers. So we managed to recruit 10 farmers for our hub, all within a reasonable distance of each other. So ranging from Perthshire, Angus and Fife. All enthusiastic, all interested to learn and all willing to share, which I'm sure everyone in this meeting is. During the initial phases of setting up the hub, it was agreed between the members what our objectives would be. So we decided that to address the challenges of our context, we would investigate new and alternative techniques, whether that's through technology or different agroecology approaches, select practices to reduce inputs and maintain outputs, and wherever possible, it should always be done with a focus on farm economics, because it is a business that's been run after all. And as a hub coach, my aim was to share the techniques and experiences to broaden the knowledge base for our members and to help them gain experience through being part of the hub. So now armed with the objectives, and this was back in 2020, 2021, coming out of COVID, we set about then our project delivery. So a programme of events was planned with some smaller on-farm meetings, which were more focused crop walks and technical demos on-farm, and also larger demonstration events with presence at a national level, where we would be part of a bigger event. So it could be in the guise of field plots or as a machinery demo, of which we did both over the years. Also, a favourite of the farmers was the opportunity to participate in cross visits to see other hubs in the other countries across Europe. We were lucky enough to have hosted a group from the Netherlands and from Ireland over the last 4 years, and also visit Italy, Ireland, and the Netherlands. So it's through this exchange that you get to learn other approaches in a different context and see actually how similar we all are as farmers. So moving on to the communication part, throughout the project delivery, the aim was to communicate IPM challenges and successes. Often the focus was on single techniques, but more discussion and debate came from linking these techniques in as part of a whole system on farm, to be considered as a holistic approach, with all of the practices on farm being considered with their effect on each other. This can be related back to a LEAF approach, which was the integrated farm management wheel, where everything is related, but also we've had examples of the IPM pyramid, and through the project, the pillars of IPM have been established as well. One thing about the project and the way that we approached it as a whole system, sometimes that hid whether the new approach was the thing that made the difference or whether there was underlying factors. And as a researcher, we're always looking for solid evidence. And we wanted to test then these individual techniques to see if they were the thing that made the difference. So could we assess the performance of individual aspects within the system, and could we do it within the project? And the answer was yes. So led by ADAS on this task, the project facilitated in-field comparisons for this purpose. Looking at on-farm trials at a field scale, we were able to test a farm standard versus a new technique in context, on-farm, and led by the farmer. With support from a hub coach, such as myself, we were able to facilitate demonstrations and provide resources, whether that was time or expertise, to test new ideas that may not have been available otherwise to the farmer, or may not have been tested to the extent that we would have. Looking at our theme today, and using BYDV as an example, I'll take you through how we identified our aims and our approach to testing in-field using in-field comparisons. So first of all, with the farmer on board, we'd identify what the aim for testing would be to help us with our trial design. So with BYDV, it could be to reduce our frequency of sprays, reduce overall incidents, and possibly if SFI is related to this, then zero insecticides, and could it be done? As a farmer-led study, we would discuss with the farmer first the potential to test what was relevant to them. So this could be a farm standard versus a decision support system, which could be the TSUM model, as an example. It could also be an old decision support, such as the TSUM, versus a new decision support, such as the ADAS designed Acrobat model. Possibility of testing early drilling versus delayed drilling, a monocrop versus companion planting, or a farm standard variety versus a variety with BYDV resistance, such as KWS Ferus or something of that ilk. One thing to point out here is that when approaching it in this way, we're not aiming to confirm what we think we know. Disproving an alternative treatment as ineffective is just as useful to us as proving a new approach is better than the status quo. Because that's the thing that the farmer will pick up immediately. They're the expert on their own farm, and they'll see the difference. So in the project, the trial aims and treatments to test were shared with ADAS Contact, and were then used to create a trial plan and provide a treatment layout. So the in-field comparisons could be done as a split field or tramline trial to compare two different treatments in field, with solutions for large or small fields within that. As it's farmer-led, the trial is co-designed to fit the farm system and the machinery available, so it can be conducted by the farmer and supported by what would be seen as the hub coach or other researchers. Large-scale trials like this are suited to assessing more transient and migrating pest populations, because they take into account landscape effects on a wider scale. Looking at BYDV, we need to take into account aphid flights and difference in genotypes, whether it's preference for certain varieties or range of flights. And things will change with the climate and topography. This can't be done on a smaller scale with plot trials, but it can be done on a wider scale, on a larger scale, sorry, with in-field trials. It does solve the challenges of spatial variation, where we can achieve more with several sites located across regions and at a field scale, and also temporal variation, with changes over time monitored with frequent assessments. What to expect from carrying out these in-field comparisons. So through conducting these type of trials, the farmer and the wider group involved in conducting these are provided with results and output with conclusions relevant to the farm in its specific context. Information may come in the form of imagery as green area index or NDVI, or even RGB if we're just looking at yellow versus green. Yield maps post-harvest are useful, and it then allows us to provide economic analysis as well, looking at inputs and outputs and gross margin for the crop. It can also be supplemented by ground truthing with in-field assessments during the growth season, yellow water traps, sticky traps, and other in-field assessments for plant health and physiology. They all add detail for statistical analysis and flesh out a final report, which informs both the farmer and the wider hub. To conclude on this approach, which I've raced through, more field trials conducted in this way across regions could build the wider picture of BYDV spread and control options, which would complement existing monitoring with on-farm validation using a farmer-led hub approach. So the work that's been done by Dan and Nasamu, this can add value to what they're doing and provide that validation at a wider scale. So thanks for your time, and I'm happy to take any questions. I'll pass back to Duncan.
No, that was really interesting. Thank you for that overview, and it's nice to see how those kind of collaborating and looking together and seeing how we can use, as you quite rightly put it, then the people on the farm and on the field, they know it best. They know their system. They know what's going to work and what's not. But you also mentioned quite well and eloquently there the importance of failure and knowing what things don't work and how we can learn from that, which is part of the reason why we've kind of developed and put up this meeting, so we can start getting a bit of a ball rolling to try and get us all discussing and communicating together in a slightly looser hub than the one you've just outlined, but to bring those lessons of what people have just tried just because they thought, I'll give that a go, and get information on those successes and those failures to help us improve home and kind of optimize strategy across different regions and drilling windows and varietal effects as well. So that was really interesting. So I'm going to actually raise one of the polls that I had, because I've realized I had several of those that we've had nice questions, so I haven't got around to yet. So if I can get Lucy to raise the polls that I had set for after Andrew's talk, I'll quickly like everyone as many people as possible to have a quick look at these and just consider, would you, after what you've heard today from all the speakers, would you be involved of getting involved, or would you be interested in getting involved in a BYDV hub that would be not a specific location, but to kind of gather people around the country to discuss about this. So those that are interested, if you let me know, we can get a kind of feel of where we're at with participation. And that's very nice to see that I can see that the main answer is a yes. That's really encouraging. Perturbed it's gone down a bit, but but now if with that one, if I can now get Lucy as the second poll that I had after this. So for those that actually said that they're either not quite interested or they might be, so people select that. So if you would be interested in doing this, what sort of time would you be willing to engage? So this would be sort of things like the meeting we're having today, or a few in-person meetings, or just time to kind of write down your kind of thoughts and methods and procedures that you've been doing. Excellent. Well, that's quite a reasonable spread with a few people that'd be quite interested in putting a bit of time in. So that's very encouraging. So thank you very much for that feedback. So if I can now ask the speakers to unmute their microphones and turn their cameras back on, we can open the forum for anyone who has any questions direct to any of the speakers, or if there's anything specific you'd like to raise that's been discussed today, please raise your hands and ask away. There was one question that I noticed that did turn up in the chat. I would like to put to Nasamu first. So this is one directed at you, Nasamu. So have you considered I'm sorry, this comes from Duncan Conabear. Have you considered in your modelling the infection earlier in the crop's development, the greater the severity of impact of the plant? In the southwest, we are already seeing BYDEB as a result of a mild winter.
Well, thank you, Duncan. Yeah. So mild winter, I think it's it's quite common these days, and with that, aphids have the chance to reproduce and stay longer and affect crop and spread BYDV. And it's interesting that it's very common in that region. So for our model, we are focused on image detection. So I can see a role there where very early in the season, in that period, we could sort of go out to see if we can detect the pest at that level to sort of control, could be effective. Again, when crops are young, there is not a lot of there's a lot of ground material, bare ground, so that could also be useful to testing in model to see how accurate it can be in detecting aphids at such conditions where the ground is more bare and there are not plenty green material. So very useful thing to do as well. I hope I've answered Duncan quite well.
Yeah, I think you did okay. I mean, what the other thing that I kind of think is interesting on that, and particularly the app that you're working on, with a couple of talks today mentioning about some of those decision support tools, those could be a really handy combination with decision support tools informing when to go and scout for aphids when you're most likely to see that flight, but then go in and be able to do a more quick and efficient search for those aphids using things like this image detection that you've been working on. So we've got another question in the chat. So this is a more general one. So this is from Georgie Bray. So full question is, we've used other IPM tools and variety choice to try and give us the best chance of avoiding impacts from BYDV. How much can we rely on tolerant varieties if we wanted to start bringing drilling date forward as an example? I suppose that would be more directed towards some of the bits that you've been working on, Daniel, or some of the students that you currently have.
Yeah, I'll just quickly rereading it. Yeah, I think it's something we need to be aware of and something we need to consider going forward, because obviously BYDV and the aphids is one part of the story, but as the climate changes and the agricultural practices change to accommodate that, drilling dates are going to be shifting. And obviously, if you're choosing a variety for virus tolerance, but you end up having to drill at a different time, there might not be a tolerant variety available. So I think it's that's when we need to start including the breeding companies and look at it from more of a kind of whole systems approach, as we need to develop all of these different things. And tools could be one way to help that. So, for example, we know that even within the tolerant varieties, is that tolerant because that plant's still infected with the virus, or that plant, or the virus doesn't replicate in that plant? Because if the virus is still present in, say, Farmer A's field, then they have a tolerant variety, but Farmer B next door is growing a susceptible variety. And is that neighbouring field then just a big virus reservoir? If there's no symptoms, that farmer might not think of applying insecticides to control for BYDV or aphids. but those aphids could still come in from the landscape, take up the virus from the tolerant germplasm, and then it could be a problem within the locality. So I think tools could be helpful way of helping address that. And I think that's one thing that we need to consider going forward, is how do all of these things fit in within the wider agricultural context, the landscape, and also the changing environment as well.
Excellent. And I think you raised the point then of kind of what you do on your farm is inherently linked with what's happening on the farm next door or within a certain radius, which is another mode of the things that Andrew mentioned about joining together in a hub to kind of work together and understand this. If we're collectively working together, we can ensure that what your neighbour's doing isn't going to affect you too heavily, and vice versa.
Yeah, and just to briefly comment on the tolerant varieties again. So we did some work last year with a susceptible winter barley variety and a tolerant winter barley variety. And we found that we got higher and more efficient virus transmission into the tolerant variety. We just didn't get the symptoms. So the aphid was able to get the virus into the plant more readily, and the virus was it was found at higher concentrations, but there were just no disease symptoms. And that might be the type of nuance that we need to think about going forward, because then, as you said, how does that affect the wider agricultural system? That was just one trial, I have to say, one small project. So it might not be representative of the reality or in field conditions, but I was quite interested when I saw that.
Yeah, excellent. And I suppose it gives me a chance to also fly out there. As part of ATB Strategic Farm East, and part of the actual reason why we're trying to build this hub, we are looking at BYDV management through the use of variety selection, and then comparing and contrasting several of the different decision support tools. But I realise Sasha just raised his hand, so if Lucy, if you can allow Sasha to unmute himself so he can ask his question.
You can unmute now, Sasha.
Thanks, Lucy. So this is just a question for Nasamu. Really interesting talk. Thank you. The app looks like potentially going to be a very useful tool as well for people going forward. I saw that you had some pretty good identification rates for the aphids. How good is the app at discriminating between different aphid species?
We have collected images of the aphids across the field and at each point. So what we have done is to test using those images that we've collected to see the performance of that model on fresh images that only have the aphids. So far, that is what the performance metrics that we've seen have displayed during the presentation. So we've not had conditions where a mixture of aphids are in one leaf and being able to sort of distinguish that. That's something we were taking forward to see again how effective can that model be in detecting and discriminating both aphids like Bachelorette aphid from green aphid. So that's something to look forward to. I also try to let you know once we get that information. Maybe Daniel wants to chip in that.
Yeah, so from what I can remember, so all the image vision aspect is done by colleagues at Sheffield University. And from what I remember from the discussions we had with them, I think last week, is, again, as Nasamu said, these are colonies where we just either have grain aphid or bird chariot aphid, but we do have an algorithm that can differentiate the two aphid species. And as Nasamu said, the next step is then to mix them together and to see, can it differentiate when there's multiple species within the image or the frame, but it can detect or differentiate between Strugnivini and the bird chariot aphid. And I think you can do it quite efficiently. I've not tested it myself yet. It's on my list to do today or tomorrow. But I think our next step is to mix them and see if it can still differentiate, however it just gets confused. But I think we're quite confident if it can differentiate them from individual images, it should be no different when they're mixed together.
Fantastic. Yeah, sounds very promising. Thank you both.
Oh, yeah, sounds very interesting. So we've got another question now from Jack. So please, you can let Jack unmute.
Jack, you can unmute now.
Hi, thank you. Really good talks. I just got a question, a follow-on question to Sasha's, which was, are you could test the model to look at Metalopendrodum as well? Or are you going to leave it at just working on the Zephyrina Venier and Dirawapaline Impadi?
I'm not, from what I know from what the Sheffield team are doing, I've only seen the differentiation between grain aphid and bird cherry oat aphid. I'm not sure if they've got rose grain aphid images in their data set. I think they do, but I can't say for certainty. I think we'd like them to have done, and I think we encourage them to, but I can't say for certainty whether they have or not. And someone might know a bit more, but from what I know, I'm not sure.
So yes, Jack, we do have a small set of images collected for rose grain aphid, but that's not as compared to what we have of grain aphid and Bercheret aphid. So that's something we'll keep on developing for the app, on the app. So
Thank you.
Excellent. Watch this space. The more things we think of, the more complicated it gets, but the more work to do and the more interesting things that we can learn. So I'm not going to get to, since there doesn't seem any questions coming straight right now, I'm going to share screen with a couple QR codes for people to follow the link to. But we've got a couple of open answer questions and surveys we'd like to put to you to try and help us build and understand what you would like to get out of a hub focusing on BYDV, so we can make it as functional, useful, and as effective as is possible. So if I can ask these you to share the closing question that I had, and I'll share my screen so people can kind of follow the links. So this is an open answer question. There's no right answers, there's no wrong answers. We're just wanting to understand what people would like to get out of a BYDV hub. But what we will also do, because realize we've had quite a nice bit of interaction, a few questions at the end of the presentations. There's a few other questions that we had that we were going to raise throughout this meeting, but we will send those across to people after this meeting. So it'd be really interesting to see what people's thoughts are now they've heard about the development and the work that Daniel and Asamu are doing, and the usefulness that Andrew showed with the power of coming together as a hub and collectively learning from our successes and from our failures. Oh, terribly sorry, didn't mean to open that on that screen. Or do that. Sorry, I just checked my emails. I've just had an email back from Basis about this, the call and points for everyone, but it hasn't. The image, I need to go into the email to find out, but I will let you all know as and when. But if you follow the QR on the screen, we've got some information on some feedback for this event, but it's also been put as a link in the chat, and also to fly it to anyone who's not aware, there is a IPM Net event coming up week after next, which we've also put the registration link in the chat. So that's a free event. I believe the morning is online, but there is a in-person component, but it's a split event. So please have a look at that, get involved if you're interested. And if there's any questions or anything anyone has after this event, or anyone wishes to get involved, you can message me directly, and I can then liaise with Andrew and Daniel on Asamu to make sure any burning questions anyone comes up with 5 minutes after the meeting, because that's normally when I come up with my questions, we can get those answered to you. But we'd really like to thank everyone for joining. I'd particularly like to thank our speakers. I think we've had some really interesting talks there that's really showing that we do have some positive work going forwards to understand and manage BYDV and kind of understand how we can adapt to changing farming practices, policy and climate. And it's also, I'd also like to give a big nod to Lucy and Bree from our team at ADAS, who've helped run this webinar and been running everything underneath the hood. They've done an absolutely great job. But that now leaves me to actually then wrap up. And as I say, any feedback would be most welcome, and we will be getting in touch with everyone who was registered for the event to pass on any of the links that we've got and to give you some of our thoughts and feelings and a bit of a synopsis of the meeting we've had today. So thank you very much, and we will close there for the evening.